From CPC, this is Biocast. Today's episode focuses on new and upcoming regulations shaping process manufacturing, specifically Annex I and USP665. To help us understand these regulations and standards and the impacts associated with each, we brought in product and application specialist Kevin Jager from Hitma Single-Use Solutions, a company with a mission to help customers reduce risk, save time, and reduce costs by delivering the right bioprocess from discovery to fill finish. And now, here's your guest host, Senior Product Manager, Michael Francis.
Welcome to this episode of the CPC Biocast. I'm your host for the day, Michael Francis. I've been with CPC about 6 years now, mainly as the product manager for the Aseptic sterile connector technology that you see over my shoulder here. I've been working on those from new product development all the way through supporting the customer with different questions they have in the field. In today's episode, we're focused on two pretty major regulatory changes within the biopharmaceutical, especially impacting the single-use or ready-for-use technologies. And to help me explain these regulatory changes, I brought in Kevin from Hitma to help us walk our way through some of these changes. So, Kevin, would you hop in and tell us a little bit about yourself and your role at Hitma?
Yeah, thank you for the invitation, and happy to join one of your podcast series of CPC. So my name is Kevin Jager, 28 years now, 3 years working at Hitma. A little bit about myself, specialized in biotechnology, process biotechnology and cellular molecular. Short history really in the R&D USP area, and now we're really focusing on implementing the single-use technology in combination with some instruments for bioprocesses. Talking about USP to DSP to fill finish, actually the whole process. And it's always a different perspective in different projects that we're working on. So it goes from really easy R&D solutions to GMP ready or bioprocess final fill applications, which are quite complex. And also one of the topics during this podcast. So, thank you for the introduction, and nice being here.
And that's neat. I think you guys have a perspective that's unique, right? Because you see so many different broad applications, right? Because you work with how many end users? You don't have to say the number, but dozens, right, of different end users across mostly the EU, or are you more globally?
So, a little bit more about Hitma. We are falling under an umbrella of a Swedish company called Indutrade. Hitma being one of those companies, and like Hitma, we have many other companies under that umbrella that are doing sort of the same thing. So we are folks in the Benelux area, and that's also where our focus area is, because we want to speak the same language, be in the same time zone, and that to assist our end customers, our relationships, the best possible manner. So we're really focused in the Benelux area. But our sister companies, our partner companies, they do the same thing, sort of.
Yeah, with all within the Indutrade group. And then for me, it was learning, I think it was maybe last year only, that so Benelux is what, Belgium, Netherlands, and Luxembourg? Those are the three main countries. Okay, you focus on. Yeah. Great. Well, yeah, again, thank you for coming, and let's dive in, shall we? So two big changes, right? Within the last couple of years, they've been rolling out, and now they're becoming more enforced. USP665 from the United States Pharmacopoeia, and Annex I. How about we start with USP665? Does that sound fair? And then go from there. Could you tell us what your perspective is on USP665, talking to customers, how's it being implemented? Just start anywhere, and we'll go and see where it takes us.
Yeah, definitely. So USP665 is a more commonly coming back question, especially since it's coming close, because it will be implemented on the 1st of May, 2026. And it started all with the FDA, of course. So I think in the USA you're more associated with it. But we slowly see the market in Europe as well to also look into the USP665 testing. So EMA, the European law, which is comparable to the FDA, they will also slowly implement the USP665 rules. So, the question is coming back, and I think it's bringing a lot of different testing and requirements from the producers, manufacturers, like CPC for instance, but also from us, the people that are assembling the product and bringing it to one of the end customers to meet their guidelines and their requirements to have the USP665 validated for their process. So I think it will be a more commonly discussed topic when we're sitting around the table talking about the different projects.
Yeah, and in the predecessor for this USP was really just an industry guideline, right, out of the now Bioform, but the Bioform Operations Group. I know most of us just say BPOG around the industry. Was that really in the EU as well? I mean, as you mentioned, USP665 is coming out of the US, but since most of these end users and the end user customers are global, these regulations end up impacting globally. And then, as you mentioned, EMA brings in regulations. That goes back and forth, similar with, we'll get to Annex I in a second, and it's kind of the reverse of that. But is that, with BPOG, was that what you guys saw as the main guideline of choice the last decade or so, or was there other regulations? Was there consistency amongst the supply base, more in this polymer single-use technology space?
Yeah, so not really. I think the BPOG was also... there was something, let's say it like that, but it was never like really a strict guideline that they had to follow. So I think USP665 now really creates a lot of stress, especially for some of the biopharma companies, that they really have to meet some of the guidelines to have a valid process running. The BPOC was here in Europe as well, but I think for BPOC it was always like a free of choice decision to implement it for your process, and when you're moving to GMP, it is a sort of a requirement. So they did it, but it was never a hard requirement, and it was always a little bit fake, testing to like the most harsh conditions and stuff that didn't always make sense. So it also limited the possibilities that you can do for your process. And I think with USP665, it is a hard one, and the difference is that it is a requirement now, a hard requirement. But I think it opens up a lot of possibilities as well, because you really get a feeling now what you can as a compatible component or material for your process.
Yeah, you're kind of mentioning the difficulty of it, and in BPOG in particular, it was kind of a guideline, and we can, I think it'd be interesting, we can talk about the history of that a little bit too. Did you see that pain more from, let's call it, so CPC, we make subcomponents, right? We don't even do any integration of systems. We sell our product over to you and others that integrate it into single-use systems, and then the end users take it on. They're the ones that really have to show that the drug product is safe. So did you see more pain coming from the end users, since there wasn't lack of consistent guidance, or from you in the integrator level or the component level, like CPC, where we had to do this testing? Or where did you see that pain really sitting, prior to 665 and the lack of clarity there?
I think the pain was really sitting at the end customer more than at like Hitma as a intermediate manufacturer or distributor. I think it always went back to either the manufacturer or the end user. But the end user, especially in GMP processes, were most of the time then outsourcing their extractable leachable studies to have it done for their process. I think for the manufacturers it was more not a strict guideline, but something to offer, like, here, see, our product is tested, we have done our validations, and it's more trustworthy for an end user or distributor to use it. So I think it was all quite fake for end users, and now it's becoming more clear with the USP665, because that's like overall for all the different manufacturers the same. It will be treated the same, and then you can specify it for your own process. So I think the impact was harder at the end user.
Yeah, I agree. And they have to figure out the leachable risk, right, to their specific drug product, which can be even more difficult. I will say a little history from the CPC side is prior to 2014, when the first BPOG guideline came out, there wasn't really any guidance. So when we were launching connectors for the biospace back in the late 90s and early 2000s, it was, okay, what do we do? And kind of the rough guidance and industry practice at that time was to say, okay, 50% ethanol for 1 or 2 time points and maybe some water for injection. Maybe you start looking at a low pH and high pH, but that wasn't very standard. And then when 2014 hit, BPOG came out, and I think we can get into this. We talked about one difference so far between USP665 and BPOG, but BPOG came out and said, hey, there's 6 solvents now: high pH, low pH, ethanol, water, and then 2 others, a salt and PS80. And then in 2020, they trimmed that guideline because it was so complex down to 4 solvents. Is there any changes you saw with people asking for different solvents or time points between, late 2010s to 2020s and leading into USP 665? Or have you seen more people, because the last couple of years the draft of 665 has been out, have you seen more people starting to look towards, okay, let's just fine tune our extractable studies towards 665? What have you been seeing through that kind of history of the single-use market trying to figure out what to do with extractables?
Unluckily, I haven't been in the market for 10 years.
Yeah.
It has been quite fresh for me, so the past 3 years, but I've been exposed to a lot of the questions with extractable leasables, but that was all, as you already mentioned, more working towards fine-tuning and going to the USP 665. So the question arose more like the validations that we have done now in-house, is that already sufficient for USP 665, or should we need additional testing? And the question always arose like the components on component level, the manufacturer, do they have it in place? So that question came up much more since the USP 665 regulation was mentioned that it will go live in 2026. So I think it went really back to, is the studies that we have already done in-house and are the processes that we are running, are they still valid, or do we have to change some things? And that is where the discussions arose, like how can we as a distributor help them? How can we have or open the discussions with CPC like you or other manufacturers of components, like what is missing and what do they have on the planning to make this happen, make it work, that you have the USP 665 testing available when it is needed for them to perform the validation on their process.
Yeah, let's poke at that a little bit. So you're kind of mentioning that the big question was, okay, a lot of us have done testing per BPOG, but 665 is a little different. So one of those differences you mentioned was on the extractions, right? Let's peel the onion back there a little bit, is what are some of those differences that you were walking through with component manufacturers or with your end users? We mentioned BPOG was a guideline, whereas 665 is enforceable. What are those other details getting into it, and how can we help the industry, talk through these differences?
Yeah, so for them it was really about the scientific justification of the threshold values. I think they're called the AET, so the analytical evaluation threshold. And that's really the number that was given out as a milligram per milliliter. Specifically for BPOG, it was always a standard number that was used. When you look at USP 665, there's much more fluctuation. So it is based on a risk metrics. I think you brought it up before as well, in levels of 1, 2, and 3, which has to do with patient exposure, but also material exposure to your product, temperature. All those things go into account with the new USP 665 testing. So that means that all the harsh conditions that have been tested, it can be less, which means it opens up positions for other components, other materials to be used as well for that process. So,
yes, that's interesting. So I think that first one there is BPOG tended to say, all right, when we look at the life cycle of a polymer tube or connector, okay, first it's sterilized. So it needs to be either irradiated, X-ray, or really gamma back in the day. And still gamma today, but I know X-ray is getting more popular. So it has to be irradiated, or it has to be autoclaved, and then it has to be placed into some kind of extraction extraction solvent. And those solvents, let's make them as harsh as possible, right? Let's make the sterilization as harsh as possible. Let's make the temperature and the solvent as harsh as possible, and then let's make these time points either, depending on it, it was, I believe it was 24 hours for BPOG, 7 days, 21 days, and 70 days. I believe there was different time points you could do depending on the part of the system you had, whether it was tubing or a bag or a film or a connector. So that was the worst case piece, right? Versus 665 allows for more customization. Is that fair to say? More, okay, what is the actual process? What is the actual risk?
Yeah, so really to specify your question, maybe to clarify a little bit, what is exactly the question?
Oh, yeah. Well, I guess you were highlighting the difference there is that BPOG was worst case, right? Whereas 665, how do you describe it? More of a risk-based approach or application-based approach? So in a way, BPOG was more harsh on the polymers than 665 is. Is that a fair statement?
Yes. Yeah, and I think that is coming back to the different parameters that you just mentioned. Like, it is a combination of time, indeed the concentration of the chemicals that you're using, but also temperature. All those things were really put to the limit. It's not that with the USP665 that you're not seeing that, but you're seeing it much more related to the real processes. So if you have a buffer bag, which holds your buffer for, let's say, a month, then it is logic to, or realistic to perform the testing on like the hardest or the longest time point for a USP665 testing. But if you have a intermediate sampling bag, which only holds the samples for maybe 12 hours or 8 hours, then it is much too overkill to perform the testing at the highest degree for the time being, because it will never be exposed in that stage at that given time. So at that point of view, USP665 is helping a lot because, yeah, as I just said, it opens up the position for other materials as well that before that didn't have a chance.
Yeah, that's really interesting. I've never really thought about that, because for our connectors, one thing that always felt like stifled innovation to a degree was that these tests are really expensive. That's something we haven't mentioned yet. And when paying for these tests, we would pay for them on a material basis, and generally highly pure resins, no kind of recycled resin, very pure, right from the resin manufacturer into an injection mold. And that restrained us from doing new, more novel materials that might give you more advantages from a chemical compatibility perspective, or a heat perspective, or a dimensional consistency perspective. So are you seeing that more, that component manufacturers or other players within the market are able to now use more materials? And do you have examples of that yet, or what have you been seeing as or is it just making it easier so that people have set testing that they can go forward with?
I think that the materials are not really changing. So of course you have your polypro, polycarb, polysulfone. I think in the past, especially from my time being the past 3 years, there have not been really new materials coming into the market. Maybe a couple interesting inventions. I think CPC also brought one in, which is a competitor for the PFAS excluded component. But you start more and more over with filters, I have to say. For components, it stayed quite the same. There's not a lot of differences going on in like really the drug contact material, which are used in single-use assemblies or in connectors.
Yeah, I think that's something that could be reconsidered now. But yeah, I agree. I think it's just for us, the end users are getting more comfortable with these set materials, like you mentioned, the polycarbs, the whatnot. But maybe in the future with less testing, we can get through that. You mentioned this conversation on, okay, is BPOG still applicable to USP665, or in what cases it is? How does that conversation go with partners that you're having it with? How do you help guide them through that conversation to seeing if it's still applicable or not?
As I just said, like, it's already a combination about it's a requirement, becoming a requirement for the FDA now. For the EMA, it's not as of yet. But you see that the biotechnology companies, they're getting more afraid that it will come slowly. It's not a question like if it will come. It's more a question like when does it come? So the only thing that we can really do here with them, because we cannot do the testing in-house. It's more on a component level. But what we do is to advise them and help them to provide them the validation guides and the information that they would need to have the USP665 testing for their process. So when we hear a customer talking about like they're having a BPOC testing or they have tested that for BPOC, we are now already advising them like it's better to already step into the USP665 world, because it will be a matter of time. And it's better, especially for GMP processes, to have that already up and running, have that validated in-house. So when that point comes, you don't have to change everything around and perform the testing like in a couple of months. It's always good to work ahead of time instead of lagging behind, and especially in the market that we're living now in with the biotech. So that actually.
Yeah, that's that is, I think, the guidance I've been seeing too, is go towards 665, and in many cases to encourage other component manufacturers out there. But the BPOG testing, since it tends to be overdone compared to the USP 665, it can be applicable in certain cases, especially depending on the surface area of the component in place. So.
Indeed. And I think it's also the rationale behind it, right? It's easier to explain it to them about the USP 665 because there's really a guideline. Like it's for every other company, it's the same. So with the BPOC, if you ask company A and compare it to company B, even though if they're, let's say, both ADC companies, it can still differ how they did the extractable leachable testing based on the BPOC. So I think it creates a lot of clarity within the market.
Especially in that world where more intense chemicals are being used, this conversation is definitely coming up again.
Yeah.
All right. Anything else on 665 that you want to pick at or talk through or ask me about, or otherwise we can transition over to Annex I?
Yeah, the question that I have for you is, how do you see USP 665 now as a guideline? Do you see it as a final stage guideline, or do you say in the coming 5 years we can expect that it will be more specified or even change a little bit in what the requirements are?
Yeah, it's a good question. I think we've been seeing, and our connectors are used globally by many different end users. So the conversation started really picking up speed in 2024. Hey, are you guys ready for this? I think one thing we were waiting for as a component manufacturer was really the final USP draft that was going to be, codified and enforceable, right? Because what we wanted to know is, and I believe some of the people who wrote the 665 and helped with it also helped with the BPOG guideline, which was what we were hoping for. Because from a chemical testing perspective, we wanted to know, okay, if we have to kick off additional testing, we need to know exactly the sterilization, the time points, the solvents, and the temperatures to make sure we can fine-tune that test so it is applicable and meets the 665 need. Or do we not have to do it because our BPOG testing, because we tested all of our current flow path materials to even the 2014 standard. So a lot of that solvent information we already had, and a lot of the same time points, because I believe, I'm forgetting off the top of my head, I think we did 7 in 21 days, or we did 1 day in 21 days. So we have a pretty good basis of data already. The last year and a half, talking to various end users and submitting our statements on our rationale behind our BPOG data and how it's applicable. For the most part, most end users have said, yeah, it looks good. You guys are giving us a very good picture, a standard set picture of the extracts we can expect from your materials based on the surface area that they are. I think the biggest sticking points I've had in walking through with customers are around, okay, it's enforceable. We got to make sure it's buttoned up. So get us this very clean documentation and very report structure, which we already had, but often it was with that BPOG template and getting it really set per customer. And then the other, I think the biggest back and forth I had was on and this is getting really into the details of it, but the C3 solvent within the USP 665 or the high pH solvent. That was talked about on, okay, the 665 calls out, okay, it should be, I think, 1 or 0.1 normal, is it NaOH? So to get a pH 10 buffer, essentially, okay, that's what we want. Whereas the BPOG called out 0.5 normal, which brings it more to pH 13. And there is some back and forth. Okay, how applicable is that solvent? Do we need to do it get an additional point, pH 10 within there or not? And it really comes down to, okay, at a component level, we're trying to give them an accurate picture of what could come out of the materials that they could expect and see, and take that risk-based approach with their leachable study and saying, hey, if this is the worst load that we see from these materials from CPC, it's not a concern. It's not a risk based on the purity of the materials. So in general, we've seen more end users accept the BPOG data as that worst case, especially in the case of the C3 solvent, and working through that. But that was one example of there's been some back and forth. With any new regulation, we'll meet it and strive for that as quickly as we can. We believe we already are. I'm sure there'll be some conversations going now as it becomes more set in stone as we go forward. But that's really what we've been hearing on 665. And then a little bit on how it relates to X-ray. But that's really a whole nother topic that maybe we could come back for on a different podcast.
A whole new podcast for that.
Yeah, exactly, on the X-ray implementation. And then any future extractables we would do if we're bringing in a new material or if we're trying to bring in a new sterilization modality, we would follow just straight USP665 at this point. So, yeah. Did that answer your question fully, or is there any other threads you wanted to pull there?
Yeah, no, definitely. I think that that is a great answer to the question that I have. And I think to add to that is what you can also see in our market, that it now came out that eventually questions will also come back from the end users, which will slowly be also integrated back towards like the Annex I, back to the USP665 regulations, and that they maybe adjust it and tweak it a little bit here and there, really based on the experience from the end market back to where it all started. Yeah,
that's, you just articulated that much better than I could. There will be some clarifications, right? And there will be some addendums needed. I think as the industry figures it out, we start answering more questions and getting into the nuances of it. So we're ready for that, but we think the data we have is looking good, and we're excited in this next chapter because I think, to your point, it drives clarity through the whole market. So that's helpful.
Yeah, indeed.
All right. You ready to pivot to Annex I, the other big one?
Definitely.
more EU focused.
Yeah.
All right. So we I think effectively went 100,000 foot level with 665 down to a 10-foot level there for a minute. So let's try to do the same thing with Annex I. So big picture, Kevin, what is Annex I? How does it impact systems? What are you hearing from the market? Let's see where we go.
Yeah. So Annex I is like, it's really broad, but it comes down to really have your process well organized, have it tracked, have the quality management regulated on a timely basis. It's really coming down to understand your process, know what's going on, and how to make it as risk-proof and reliable as possible. So that's it really in a nutshell. But when you break open that nutshell, it opens up a lot of possibilities and things to focus on what to do with the Annex I. And there it comes back a little bit with what you have with USP665, which went really clear based from BPOC. I think with Annex I, it's still moving between a few layers. Like there's multiple different options how you can solve things, and some things are still not like really clarified. They're still in a gray area. Talking, for instance, about the sterile connection. So there's more movement over there, and I think the Annex I will, over the coming years, will be specified more and more often. I think it will be more strict or becoming more strict than the USP665, because you're starting at like a level which will be going to the same level at a certain point.
Yeah, so let's talk about that, and let's maybe back up for a second. So Annex I is out of the EU. What regulatory body came out with it exactly?
With the regulatory body, you mean the EMA?
Yeah, it was at EMA that wrote it. Yeah. Okay. And then you mentioned around the design of systems, and is it is it as enforceable as 665, or how does that enforceability work with Annex I? As a U.S. manufacturer, it's not as clear for us, right, as stuff coming out of USP.
So it really depends. Of course, there's already like a strict separation between already existing processes and new processes. So that's something that you see in the biotech, like if something already exists for the past 10, 20, 30 years in drug manufacturing, as long as you don't break it open, it's still a valid process. Mm-hmm. So for new processes, Annex I, like what you currently see is that it is changing quite fast. So the Annex I that you had 1 or 2 years ago, it will be slightly different than the Annex I that you see now with the regulations that they focus on. But it's also person specific. So if you get an inspection based on your process, what works for someone at company A, it can maybe also work for someone at company B, but it will not be like validated. So you have to be really good in providing the data, the decision why you make it, the arguments, and if that is in line. So it's more about the quality management that you have and contamination strategy control. If you understand your process and you can argue or give the arguments why you're doing something and it's valid, then it's okay. But that's still a little bit the gray area that I'm talking about. So they give some options, they give some things to think about, but it's not really specified that you must do it. Like it's not a hard requirement.
All right, so let's dig into that a little bit deeper. So you mentioned Annex I helps with your quality management, helps with your kind of risk mitigation around the sterility of your process. What else does Annex I do? And you can take this from what behaviors does it encourage from the end user, but also the system design? What are those key areas that Annex I of the process, those key areas of the process that Annex I is really looking at saying we need to control or have a control plan for this part and this part? Is there some key areas that you're seeing it being applied to?
Yeah, so it's really, I would say, about understanding the risk of contamination in your process and how to get that down to the lowest as possible. So when I'm saying that, I mean, how can you make sure that your process from step A to all the way down to step B is a valid process that you can trust? So what you want then ideally is as minimum connections as possible. And if you make a connection or a disconnection, to do it as aseptically as possible. So then when you're looking at the process, it's really pushing customers at overthinking their current design, making things more compact, think about more efficient assemblies, transfer lines, and don't make connections which shouldn't be there or don't add a lot of value. So it's really bringing efficiency into play when you look at the production process at the moment.
Yeah, bringing that closure of the system too. Is that a term that's used a lot within the EU or Annex I? As far as we talk about it, right? With connection technologies, we make open connectors. Those are the quick connects, the MPC fittings that people are familiar with that came out in the 1990s. But then we have closed connectors or sterile connectors that have the membrane on them in place, like the AseptQuick brand. Is that open and closed terminology used a lot in the Annex I conversation, or is it more non-sterile and sterile, or what's the words that are often used?
I would say more, it's a combination about aseptic with sterile and non-aseptic, non-sterile. Open connections, it's when you divide the process in two, right? So it isn't mandatory to have aseptic connections or closed connections throughout your whole process. So when you're looking processes before a sterile filtration step or a filtration step in general, it can be open. That's not a problem. And even nowadays with new processes coming up, you can still have open connections as long as the step where your product has to be sterile at a certain point. Then you have to claim an aseptic or a sterile process. And that's where you are looking at like the aseptic connectors from you, look at overmolded capabilities, welding, sealing. So it's really a give and take, and it depends on who you're talking to and at which step of the process you're looking at.
Yeah, you mentioned that line is drawn, right, on and where that line gets drawn of, okay, this is where the sterile process needs to take place. A big part of that is, and I know you talked about it a little bit when we were in our prep work for this, is around pup sit, which I always think is a command for a dog trainer, but what it actually stands for is what you can check me here, what pre-use, post-sterilization integrity test. Is that?
Yes.
Yeah,
you're
okay,
right.
I think I got it right. Yeah, okay. It's quite a long acronym, but so the pup sit process, how does that interplay with Annex I? What are you seeing from the filter manufacturers or filter design setups? How does that play into all of Annex I?
Yeah, so PubSit is, I would say, one of the most complex implementations when you look at the Annex I. Before that, you, of course, had your filter, which is manufactured, it's tested, you have the documentations in there, and then after that, it will be sterilized, used in your process, and then back in the days, it was always used as a post-integrity testing. So after your process has been completed, your drug product, which can be a couple of thousands to a couple of millions of euros, the whole process is done, it's filled, then the filter was tested. And 99.9% of the time, the filter passed the test. But there were incidences that the filter didn't pass. So then you have two outcomes. One will be to go back to the manufacturer, and they will do additional testing. And then from that 0.1%, still like at least half of the filters were still passed. So you were able to continue. But there were also incidents that your filter didn't pass. And then you have the problem. Because, as I just mentioned, you can have a drug product which is a couple of thousand euros, or you can have a drug product when you look at cell and gene therapy, or maybe some ADC products, which go from a couple of 100,000 euros to a couple of million of euros. And that's where you preferably want your filter tested before you use it. So PubSit created that possibility. It's an intermediate where the manufacturer tests it, you test it post-usage, but now you can also test it in line pre-usage. So after sterilization, there has been an impact on the filter, but still before you run your process. And that's where you have a bubble point test or a forward flow test. Forward flow is more perfusion. Bubble point is more reaching the limits with a sort of pressure until it bursts. And that's where you test the integrity of the filter. So you really specify that the couple of minutes or hours before you use the filter, it's installed, sterilized. Is it still functional? Can we trust it? And then if it's not working, you can always remove it and then click a new filter in there, because that couple of €100 for an extra filter, an extra assembly that you interchange, it saves you down the road a lot of money.
Yeah, that's the cost of failure, right? Near the end is so high that often we see that extra testing come into play. What advice are you giving end users, or what questions do they have around Annex I or PubSit that you could give guidance on, or you've seen as trends within the industry?
Yeah, so I think Annex I is quite straightforward. They're coming back with the questions like, how can we make the smart design for your final fill, and how can we make the aseptic connections as trustworthy as possible? I would say it's more complex on the PubSit part, where we have quite a close role with our end customers to help them design such a process, but also to think along like, what are the capabilities that you can do with a PubSit? Because the setup of a PubSit, if you look outside of the filter, you need a venting line, you need a line for either your water, your buffer, or your product, because it's 3 different methods. Do you want a sampling bag? Do you want a sampling bottle? Do you want a combination of a water or air filter? There's different possibilities with the PubSit, how you can design it. So we always get the question back like, how can we design this based on the process that we have? How can we make this as efficient as possible? But also, how can we make it as compact as possible? Because you have to imagine, sometimes with like a 20 or 30 inch filter, you're talking about a shadow wall of 1 to 2 meters. So it can be a quite big assembly or process step that you have to implement in your process.
That's fascinating. That's a lot of intentional design work that needs to be really thought through up front. On Annex I, as we wrap up here, single use has been coming out more and more last 20 or 30 years. How does the use of disposable or single use technologies interplay with Annex I? Is there any overlap? Is there any guidance you've seen there or trends as well?
Yeah, I think single use provides really a good solution for Annex I because, as I just said, it also goes back to quality perspectives. When you're thinking about multiple use, you have to take into account that you have to track the sterilization. You have to track whether there's no contamination risk and stuff in your process. How is the process ended? How does it start up? It's all parameters that you have to measure. When you look at single use, it answers a lot of those questions already for you. So when we come with an end product which is completely sterile and ready for plug and play at the end user, they only have to unpack it 1, 2, or 3 packages, and they can install it because we, together with you as one of our suppliers, make sure that they have all the documentation available. It's sterilized in the manner that it falls between the gamma irradiation doses. So all those things on quality management and the contamination risk that they have to take into account, single use answers that already for you. So for an end user, it makes life much more simple. And of course, beside that, when you think about like the economical side, you don't have a downtime. So I would say that single use for especially Annex I, it will become more and more interesting for end users. And that's also what we see in the market, right? So that's also a question that I have for you later on. Like, how do you see the development of single use? Because when we look at the Benelux, I think especially for new projects that are set up, it's not even hybrid anymore. It's like really 95% single use.
Yeah, so yeah, let's take that 1-2 punch here. So the first, in wrapping up maybe Annex I conversation, so single use is helpful for quality management because it makes it so you don't have to do a cleaning validation or have the downtime between there. And then 2, it's just ready to go, ready to use. Is that fair on kind of the key reasons why it could be helpful?
Yeah, definitely.
To sum that up well, yeah.
Yeah.
And then your question was around just single use in general, what we're seeing and what we're hearing and things like that.
Yeah, because you also it's less that we collaborate with, but you also have like the industrial side, of course, which is more stainless steel and things not used in bioprocessing. So you maybe have a combination feeling in that. But how do you see the development or the demand of single use in the biotech, the biopharm sector?
Yeah, I think it's interesting from a CPC perspective. Back in the mid-2000s, we released a product called the Steam Through, which kind of bridges the gap. The purpose of that product was that you could hold a steam line to a stainless tank, but it was a polymer, essentially a three-way valve that once you actuate the valve, you could run it and attach it to a single-use filter. So we've seen that bridge in the rolling out of single-use. There's a lot of articles now written on the advantages of single use, but I think one interesting one that we've seen is that capital risk is lower with single use because you can put in a new site or new process much quicker. So you don't have to worry about the payoff period of a large stainless site coming through, which has been just something I never thought about. But I think that when you look at a capital investment from a large pharmaceutical company, it makes single use a little bit more attractive there. The main thing, even back in my days, I used to work for Emerson, and when we looked at it there and here is that single use, and you already mentioned it, is it eliminates essentially batch-to-batch contamination or the risk of that because there's no more cleaning. You use a new set every time. So that's a big piece of it. And then flexibility as well. I think as the world trends away from more general medicines for a large population to more personalized or personalized to a specific patient group, you need smaller batches, right? And that doesn't really work super well with stainless because you have set tank sizes. You can't just cut a stainless tank in half and then go. But with a single-use ballroom suite, you could run a 1,000-liter batch for a while, then drop to 100-liter, then go up to 2,000-liter. And you can do all that within the same kind of space and footprint. So that advantage is there as well. I've been seeing, just more and been watching for more consideration and really holistic view of just the environmental impact, though. I think that's something that the single-use market needs to answer and continue to answer better and better is how are we, okay, we've made it work. The process is clearly working now. How do we make it better? One, for just efficiency. So think Annex I, more closed systems, less risk to patient, but then just also less overall damage and use of plastics within our world. I think minimizing that just from a design perspective. I know the amount of plastic we use is a tiny, tiny portion of the whole global use of plastic, but I think we should be really thoughtful as a market too, is like, how could we have a block of plastic, can we core it out and then eliminate our plastic use by 30%? And just start doing things like that where we think thoughtfully about our design, not just from the use of it, but also the design aspect of minimizing that impact. So I see it growing more from the capital investment perspective and the elimination of risk batch to batch. Any thoughts there of what you're seeing in the Benelux region or any questions with that?
Yeah, no, I think you mentioned two very interesting topics, right? So first one that you touched upon was really the volume size. So indeed, when single use came into play 25 years ago for the biopharma market, stainless steel was really the standard thing. Your stainless steel piping going from a quarter inch to 3 or 4 inch. You had stainless steel tanks going from 100 liter to a couple of thousands to 10,000 liters. And that's a development that you see in the market now, right? So it started off with the standard sizing around half an inch, a quarter inch. And now you see in the past years that for the pumps, for the connectors, for the tubings, but also the bags, it's going from really small sizes. I think connectors now go to 1/16th, being the microscenics one of them. That's insanely small that you're talking about, that can be realized with single use. But on the other hand, you're also going to 1.5 inch, maybe 2 inch size aseptic connectors or open connectors. And the same goes with bags, bottles. It goes all the way from 60 ml till I think the biggest single-use bags that you use now for bioreactors are 6,000 liters. So at a certain point, there's no limitation anymore that stainless steel can offer and single-use can't. So it will be more and more that single-use can replace the whole stainless steel process. So that's a very interesting topic. And then the other one that you mentioned about sustainability, I think it depends a lot where you're located. Talking about if your biotech facility or production facility is in Africa or Europe or America or Asia. But there have been more and more studies going on in sustainability. And as you know, single-use plastics, it sounds dirty. But when you think about the water, the electricity usage and the contaminated water, that's associated with the cleaning of the stainless steel processes that you have. I believe that all the way on the bottom, when you make the calculations, the difference is not that big. It really depends on a lot of parameters that also come into play. But when you compare stainless steel with multiple use, it's not too bad when you look at the sustainability aspects.
Yeah, I think we should have another podcast on that because obviously, I mean, CPC on the table, like we sell more to the single use market, so there's a bias there for sure. And I think there are some areas where stainless makes sense. We talked about, up to 6,000 liters, seeing some of those bioreactors come out. But if you have a super standard medication or pharmaceutical product that makes sense for a large population, stainless might still make sense in those cases. But yeah, I think that would be a good thread to pull as well. I mean, I have an engineering background, so you look at life cycle analysis, right, is the key word there. So looking at, steam generation and chemical use and all that. If you're, like you said, in an area where there's water scarcity, using a bunch of water to clean stainless tanks may not make sense. It's just something to consider and think about beyond a simple calculation that, any plastic we're throwing away is bad or any chemicals we have to use for cleaning is bad, and looking at both sides of that. So I think, Kevin, let's maybe take that one and have a future conversation about it. And especially we could even do some reviews of different articles that are out there. I know BPSA put out one and a few other organizations have done as well. So on the sustainability of single use in the research behind it. So thank you, Kevin. I think we'll wrap up from there. So USP665, major change just from the guideline of the extractables world to more of the regulation, and then Annex I, helping to drive clarity on, okay, how do you manage the quality and the risk mitigate of your sterile process and ensure it's sterile. Really interesting conversations, and thank you for your expertise and your knowledge coming out of Europe and talking to us about this today, and hope you have a wonderful week and holidays coming up here soon.
Thank you very much for the invitation. I really enjoyed it. Had some good conversations. And yeah, as we said, I think we already touched upon 2 or 3 extra topics for the future planning. So yeah, let's keep in touch. Enjoy the weekend. And yeah, it was nice meeting you again.